Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award-winning Ozcast Network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast.
Ozcast.
Simply head to ozcastnetwork.com for details.
Welcome to This Medical Life podcast. These are the stories of medicine with Steve Davis and Dr. Travis Brown. This is the story of humans and Neanderthals.
War. Thank you for the introduction, Steve. So we are talking about humans and Neanderthals, and I've always found ancient history fascinating. And this is relied upon traditionally in ancient texts, kings, queens, people, famous battles, and it's written history. But we're now in an era, modern era of understanding ancient history through genetics and mixing of different populations and looking at DNA for us to learn about human species and the different variations. The amazing thing about DNA is it is a difficult thing to work with because it degrades.
Yes.
So we know that some of the oldest DNA that we've had in 2013, there was an ancient horse DNA which was sequenced. They said was about 500,000 to 700,000 years old. In 2021, there was a mammoth found in the Siberian permafrost, which was over 1 million years old. The oldest DNA that we actually have is a Neanderthals, which is estimated at being about 400,000 years old. And so it's just an incredible sort of part of human history that our understanding is that the last human and Neanderthal common ancestor is about 500,000 years old plus.
And this has pertinence for this medical life because our medicine is trying to work with this genetic tapestry.
And this is part of our understanding of everything, where we have come from. And we understand that Homo sapiens, so what we call humans today, came out of Africa about 100,000 years ago. But it's likely that Neanderthals left Africa and evolved outside of Africa well and truly before then. And so that we have been able to sequence the full Neanderthal genome, and this was done in 2014. It turned out to be a female from the Alta Mountains in Siberia. It seems to be that the human population, or the early modern humans, had a much broader population of genetic diversity, whereas Neanderthal was much smaller and I think in pockets of isolation. And so they showed high rates of inbreeding. And this is about, what does Neanderthals look like? Well, our understanding is from the fossils that they had short limbs and torso, but broader noses. The first fossil we found of Neanderthal was in 1829, but it wasn't known to be Neanderthal until about 100 years later. And so the first real fossil that was called Neanderthal was found in the Neander Valley in Germany in a Feldhofer cave. This was 1856. And recent studies have suggested that there is a crossover period about 50,000 years ago of intermixing and interbreeding between Neanderthals and humans, and this spanned a population of 7,000 years. And so it's pertinent today because we understand. What I'm trying to work out is, like we've said, that there's a certain percentage of human DNA that has Neanderthal genes or DNA sequences in it. And so that I sort of found fascinating, and it's part of where we've come from, our rich history, and trying to work out where does that come into it today. And so this is more of an interest as to where we've come from and what it means for us is today.
I wonder what we'll learn about ourselves as we get into this interview. Associate Professor Bastien Yamas is a former ARC Future Fellow and is affiliated with the Australian Centre for Ancient DNA. He's also an honorary research associate in the Indigenous Genomics team of the Kids Research Institute Australia, honorary associate professor at the National Centre for Indigenous Genomics at the Australian National University, and Deputy Director of the ARC Centre of Excellence for Australian Biodiversity and Heritage. He aims to integrate past and present Indigenous genetic diversity from populations around the world to understand how humans adapt to their environment. And Associate Professor Bastien Yamas is our guest on This Medical Life podcast. Welcome.
Hello.
To start the ball rolling on this fascinating topic ahead of us, I'll start with an easy question. How many different types of human species have been identified?
Yeah, so just as a disclaimer, I'm not a paleoanthropologist. I'm a molecular biologist by training, and I fell into that field kind of through like some connects research, but I find it very fascinating. Human species, the definition of species is probably something we're going to talk about a little bit during this interview, but there's probably 7 or 8 human species that have been identified so far. There's us, what we call the anatomically modern humans, Homo sapiens. And then as part of the genus Homo, there's a couple of other cousins. Couple that people might know about already, like Neanderthals, Homo neanderthalensis, which was found first in Germany, in the Neander Valley. But then we realized very quickly that there were Neanderthals pretty much everywhere in Western Eurasia. And then there's another elusive cousins that we call the Denisovans. They're not a species per se, because we virtually have so little in terms of fossil remains that we don't even know really what they look like. But we do have a genome. We'll talk about that a bit later. And they're sufficiently different from Neanderthals to really call them a different, at least subgroup or subspecies, if not a species. And they come from Siberia. So it's most like East Eurasia, if you want. And then there's a flurry of, a handful of other homos, Homo floresiensis in Indonesia, that we also call or nickname the hobbits, because they have a very, very small size. There's Homo heidelbergensis, a few names like this, which is a little bit older, got extinct a while ago, 300,000 years ago, as opposed to the ones that I just talked about who, got extinct 30, 40,000 years ago. And heidelbergensis was found in Atapuerca in northern Spain, in a very famous fossil place. There's Homo habilis. Oh, sorry, I'm trying to list them in my head, but there's quite a few. So Homo habilis, again, very old cousin, got extinct very, very early on. That's the Like family. We found them in Eastern and Southern Africa. Homo naledi, that has been identified very, very recently, a couple of years ago already. And again, an old cousin from South Africa. And I'm probably Homo erectus. Which is also someone that people usually know about. Homo erectus was very widespread across Asia, Europe, even Africa. There's a couple of famous Homo erectus, like the baking man, for example. There's also the Java man in Indonesia. And there's a site in Georgia, Namisi, which also has, a little bit of reputation in newspapers usually. So you can see that there's quite a few Homo species that have been identified so far.
How does one classify different human species?
My colleagues who work in that space, basically what they use is fossil remains. And the problem is that the fossil record is very patchy, not only through time and space, but also extremely fragmented. It's very rare that you're going to have a fully articulated skeleton for you to analyze. Okay, most of the time, you're going to have parts of the skeleton, and then even those parts might be completely fragmented. So it becomes a game, a puzzle, basically, to try and solve what's going on here. So observations usually are focusing on teeth and craniofacial morphology. You can also use what we call postcranial morphologies, anything that is not the skull, basically. Yeah, craniofacial morphology is really what is used a lot. And then from there, people will have a look, measure, basically. They will have a look at the shape, the size of molars, canines, incisors. So that's for the teeth. And then they will look at how the mandible is, its size, its robustness, how it's attached to the rest of the skull, and then the general cranial morphology as well. And cranial morphology, of course, links a little bit to brain size as well. So there's a lot of discussions around cognition and the size of the brain, what makes us human and what makes our cousins human as well. And there's probably, in terms of the ease of understanding of differences, basically, we could compare Neanderthal and humans, for example, anatomically modern humans. When it comes, because also for Neanderthals, we have quite a lot of fossil remains. It's quite interesting because it's, like I said, Western Eurasian, usually, and the climatic conditions over there are relatively good. And the extinction as well of Neanderthal, like 30 to 40,000 years ago, is young enough or, late enough that we also have quite a few remains left. So we do have those entirely articulated skeletons. And that's, that's a bounty for paleoanthropologists. They have all they want to do, basically, to do the comparisons. So if you look at the cranium of a Neanderthal and a human, humans will have this really round-shaped skull, whereas the Neanderthals the skull is going to be rather elongated. And I'm talking about averages here, okay? There's variation within the species, of course. And then there's going to be a lot of differences with the molars as well. So Neanderthals will have slightly bigger molars, flatter as well. Going back to the skull, there's going to be a pronounced brow ridge for Neanderthals as opposed to humans. There's going to be a little bit of prognatism as well. So the face is elongated compared to the flat face in anatomically modern humans. And what else? There's a chin bone that is more pronounced in Homo sapiens as opposed to Neanderthals. So you see, there's quite a few features that, even if you're not a paleoanthropologist, I guess that if we were giving you a holotype of Neanderthals and of Homo sapiens, you would probably observe the differences yourself. Then it becomes a bit complicated with the other human species. And there's a lot of debates, as soon as a paleoanthropologist identifies a new species of humans, I can tell you there's a lot of colleagues who are going to scrutinize that very carefully. And then there's a lot of claims that have been made and then revised. And it's a very dynamic field of research, if you want.
Now, there's a lot of discussion about the crossover of Neanderthals and humans breeding. There's a crossover of history. What do we understand about that relationship today?
Yeah, that's an interesting one. So there's a crossover in time, but also in space. And the definition of species usually says that two different species will have some sort of reproductive barrier. Meaning that if a member of species 1 meets a member of species 2 and they reproduce, there might be some offspring, but those offsprings usually are going to be sterile, or there might be no offspring at all. It's just lethal in the womb, and you don't talk about it anymore. In the case of humans, it's quite interesting because those cousins, Neanderthals and Denisovan in particular, it turns out that we have evidence from the DNA that there has been interbreeding between Homo sapiens, Neanderthals, and Denisovans. And not just only one way, okay? It's going to be both ways, basically, and between the three. Of course, I mean, if you just have a look at the bones, it's going to be extremely difficult to make that call. Although, I mean, some people, like late in the 19th century, Thomas Huxley, for example, which is a famous evolutionary biologist, they had already hypothesized that there would be some interbreeding between Neanderthals and humans. So that hypothesis had been going for a long time. But it's only when we managed to extract and analyze DNA from ancient humans and modern humans as well, but especially Neanderthals and Denisovan, that we realized, oh, actually there's signatures in the genomes of people living today of Neanderthal and Denisovan genetic information. So it does exist, and it's a proof basically that something happened, interbreeding happened. The frequency of it, where it happened, when exactly it happened, that's, again, a very active field of research. There's a lot of papers pumped out every year over the past 10 years, but we have the evidence that it happened.
When we think of it simplistically, species 1 and species 2 shouldn't really develop offspring, or they might be sterile. The fact that this did happen, is that because even when we define what is a species and what is not, there's a continuum and there's an arbitrary point where we have to rule off, and so that does allow for them to be close enough for this reproduction to happen?
Yeah, I mean, we could have a very long philosophical question, but
sorry.
In biology, species has been defined many times. And in anthropology, the definition of species will be based on morphological observations. Okay, so you don't have access to those offsprings. You don't have access to the possibility of two individuals mating or not, because, well, you're looking at fossils. So except if you have a time machine and you're able to go back and, see those two species in the act, it's going to be a very challenging task to be sure that they are species. But there's enough morphological differences that and not only differences, but also it's something that is consistent across, multiple observations. So something that makes you relatively confident that you're observing two different things. But then you move to genetics, and genetics, there's other definitions of species in that space. You need a number of differences in the genomes of two organisms to determine if they are species or not. And I tell you that there is no magic number. Okay, I mean, there's no agreement on exactly how many differences you should observe between two genomes to determine that it's two species. And then after that, you have the purely biological definition of species, which is going back to this act of, interbreeding and then offspring being born or not, or surviving and being fertile or not. So a range of definitions, basically. But you're right in the sense that this continuum seems to exist. And there's no clear-cut speciation event, basically. It's something that happens over time, over many generations, many iterations of changes. And eventually, at some point, you will end up with two organisms that are so different based on their morphology, based on their reproductive system, based on their genome and the structure of their genome, the information content of their genome, that there's impossibility of even mating, or there's impossibility to have any offspring born from that mating event.
You mentioned before that some of the genes are still recognizable. Do we know what the Neanderthal genes are and do in humans today?
Maybe I'm going to touch a little bit on the development of ancient DNA research, so what we call ancient DNA, as opposed to the DNA from living people, basically. And ancient DNA is a fascinating field of research. I mean, I fell into it 16 years ago, and I never stopped. Coming from a biomedical background, okay, I had no idea about this field of research. But basically, ancient DNA is the DNA you retrieve from the fossil record, from long dead organisms, basically, including humans. How ancient is ancient? Well, it's variable. It depends on climatic conditions. If you have something preserved, above the polar circle in what we call the permafrost, the soil that is permanently frozen, then you can go quite far back in time. I think the oldest ancient genome at the moment is from a mammoth, and it's roughly 1.4, 1.5 million years. So we're going into the million years there, okay? But we're certainly not going into the 60 million years, back into the Jurassic with dinosaurs and everything. So need to be careful there. There's a limit to what we can do with ancient DNA. But to be fair, most of the ancient DNA analysis are usually in the range of a few thousands of years, a few hundreds of years, down to a few tens of thousands of years. So we're roughly talking about the last 100,000 years, if you want. And that allows us to analyze DNA from fossils from many places around the world. Usually you'll need some special conditions though, caves are great, stable environment, relatively cool over time. That's a good place. Some special environments where there is no oxygen, where the temperature is stable, will do as well. Bugs, for example, in the Netherlands or in the UK can work. There's also some randomly well-preserved cemeteries or burial sites, that you may find around the world. So anyway, ancient DNA is tricky though, because it degrades over time. Imagine you have an organism dying. Once that organism is dead, there's no molecular processes in the cell to maintain the integrity of DNA. So slowly over time, DNA is going to be chewed up by bacteria, because that's a great source of energy for some bacteria. It's going to be broken down through biochemical processes. UV radiation is going to do some damage to it as well. In essence, you're going to have shorter and shorter fragments, and those fragments are going to be damaged. There's some bases, you remember A, T, G, and C, that's the 4 letters of the DNA alphabet. Some of those are going to be changed into something else. So that complicates things, because if you have a beautiful skull of Neanderthal and you manage to extract some DNA from it, well, first, you're going to have a lot of bacterial DNA invading that skull coming from the soil, okay? So the amount of Neanderthal DNA is going to be tiny, tiny, tiny in comparison to what we call contaminant DNA. And then that Neanderthal DNA might be contaminated by the human archaeologists who found it as well, which is a problem. And then it's going to be fragmented down to very short pieces, okay? The human genome is 3 billion of those letters, A, T, G, and C. But those fragments are going to be something like 10, 20, 30, maybe 50 of those letters. So reconstruct the puzzle, like using pieces that are 20, 30, 40 base pairs for reconstructing something that is like in the billions of them. It is challenging. And then there's a little bit of damage as well, which means that some of the letters that you read in the alphabet of the DNA, in particularly C, are going to be changed into something else that results in your sequencing data into T's. So you observe a T, but originally it was a C. And you can observe a mutation C to T, which is actually not true. So it is challenging, but we do have ways. I mean, over time, we're talking about 30 years of development of that field of research. So over time, we had help from technology. Sequencing technologies have improved dramatically. We can sequence now a lot for not a lot of money. And then we also have computer sciences helping us a lot, because this ton of data that we generate can be computed and we can make sense of it. To finally reply to your question, I'm sorry, it was a bit convoluted, but yes, we do have access to Neanderthal genetic information. We actually have some very well preserved genomes that have been sequenced and for which we are absolutely confident. It's the quality that we are comparing to people using genomics in medical research for present-day people. So very confident about the sequence of these particular individuals. And looking at that information, we can then compare it apple for apple, basically. Same place in the genome, same genes, and then we can start looking at, okay, so what are the slight differences that we may observe between Homo sapiens and Homo neanderthalensis? And there's a lot of them. There's a lot of them because, yes, there's been a split between Neanderthals and Homo sapiens probably 500 to 600,000 years ago. And for a while, Neanderthals have evolved their way and Homo sapiens have evolved their way as well. And that during that time, there's been an accumulation of differences, but there's still enough similarities, basically, that we can literally call them cousins. And most importantly, there's enough similarities from the structure of the genome, the number of chromosomes, and then the content, the information content as well, that we could have babies. And those babies were very healthy and they kept reproducing over time. And it ends up with us today. If I sequence my genome, it is very likely that I'm going to find chunks of information in my genomes that come directly from my Neanderthal ancestors.
And is there an example of what some of those chunks how that would manifest in you? What would we notice?
Not much at first, okay. To be fair. But it's really hard to detect the Neanderthals in us just, looking randomly at someone in the street. What I may want to say at this point, though, is that this Neanderthal introgression of DNA into humans happened once humans left the early Homo sapiens left Africa. So usually in people of African ancestry, you won't find the Neanderthal information. You will find it in people everywhere else in the world, basically. And then there's also the Denisova that I mentioned earlier, for which we also have a very good quality genome, despite the fact that, you remember, I said we really have just a few pieces of bones and teeth, but we still have a genome. So this is the first species, if you want, has been identified from the genome without having any idea about the skeleton. Anyway, this Denisova integration happened mostly in our region of the world. So Papua New Guinea, Aboriginal and Torres Strait Islanders, Australians, and then East Asia, so island Southeast Asia, a little bit of Oceania as well, and then South Asia. So there's a little bit of a mix of Neanderthal and Denisova in this region of the world, whereas Western Eurasia, it's mostly Neanderthals. That said, so people have looked at human genomes, Homo sapiens, you and I, living people, and then they started to catalog all these regions of the genome for which they could find some Neanderthal information. On average, I will have 2 to 4% of Neanderthal information scattered across my genome. Okay, so it's not going to be big chunks of it, like an entire chromosome. It's going to be tiny pieces here and there. And then, my brother will have relatively the same proportion, or relatively the same content. But then people, living in France, where I was born, they will have a range of that information across the genome and not necessarily sharing it. Okay, so everybody carries 2 to 4%, but not exactly the same 2 to 4%. Meaning that if we were combining millions of people carrying Neanderthal DNA, we might be able to reconstruct the entire genome, or close to it anyway. Except that for some parts of the genome, like chromosome X, for example, there's entire deserts of Neanderthal introgression. It's been purged over time. You may think that, it's negative selection. Something happened across generations where people carrying Neanderthal information on the X chromosome, simply the baby were not born or could not reproduce, for example. But that happens as well across other chromosomes. Okay. So there is a process in place whereby it seems that some of that information is purged over time, but still it exists today. If it was purged, I would tell you, 40,000 years later, there would be nothing left in our genome. That's how selection works. But we observe it, so there's also positive selection. Some of that information is retained over time. And then people had a look at that, and then they realized, damn, that information plays a role in metabolism, in cognition, in immunity, in skin, hair, and eye color. So there are actually external traits, my skin color maybe, but also like internal traits that you may not be able to see straight away, but maybe I have a propension to develop type 2 diabetes or a liver disease, later, that may be driven by a Neanderthal genetic variant. So we are all shaped basically through generations of admixing between Neanderthal and Homo sapiens. Then the Neanderthal got extinct, and the humans kept on carrying that information over time, up until today, where there's still some of that information that impacts probably our health and disease.
There was an article saying that our impression of Neanderthals is not the dark-haired, sort of dark skin, but actually red hair and pale skin. Is that right?
Yeah. So we're going back to 2007, I think. Don't quote me on that one. But colleagues in Europe, they managed to sequence just one gene, MCR1, melanocortin receptor 1. And that gene is involved in the determination of skin and hair color. And what I found out is that by looking at the variants in that particular gene from 2 Neanderthals from Spain and Italy, the information or the function of the gene that I could infer was that there was a loss of function in that particular gene. So basically, it was not functioning very well in those 2 particular Neanderthal individuals. And what we know from the study of the same gene in present-day humans is that people who have this exact same loss of function of that gene, so they inferred that in these particular Neanderthals, those 2 individuals, they had fair skin and ginger hair, basically. The fact is that when people started to look at other Neanderthals over time, they realized that not all Neanderthals carry that MCR1 variation. And so there is a huge variation of skin color, eye color, and hair color in Neanderthals, the same as you would observe in Homo sapiens. So this characteristic, representation of Neanderthal that you might find in some museums around the world, actually, of red hair and fair skin, and usually blue or green eyes as well. That is probably based on a very atypical genetic information that was found in 2 individuals, but it doesn't represent the whole of the populations plural, probably even, of Neanderthals.
Wow. Well, thank you for that. I'm going to take a moment to reflect on that, because looking at Dr. Travis Brown, there's sort of a reddish tinge to your hair and blue eyes, and now I'm seeing him in a new light. We'll come back in a moment and we'll shine our focus here on Indigenous Australians. Let's continue our discussion with Bastien now. Bastien, you've also researched the genetic history of Indigenous Australians. Can you tell us a little bit about that?
Australia has been isolated for a very long time from the rest of the world. And so a disclaimer, okay? I am going to tell a story through the Western scientific lens, okay? And I do respect the traditional views of the different First Nations of Indigenous Australians. And they do have some very clear understanding of their origins and the story of their ancestors, okay? So the story that I'm going to tell now is based solely on anthropology and genetics. So basically, what we understand of the evolution of humans, of Homo sapiens, is that they evolved in Eastern Africa. And then at some point, they left the continent. Of course, some people stayed in Africa and kept on evolving in Africa. But this diaspora of people leaving Africa and then slowly going around the world, in the very early times of that exit from the continent of Africa, people managed to go through South Asia, island South Asia, and arrived in what we called the continent of Sahul, which is basically a landmass connecting Papua New Guinea and Australia. So the sea levels at the time were so low because of ice sheets covering the North and the South Pole, that the land of Papua New Guinea was actually connected to the land of Australia. The strait in between the two islands today is very shallow in a sense. So there was this gigantic landmass, and then people reached this place very early on after leaving Africa, and then they remained isolated from the rest of the world. So they carry with them some very old ancestry, okay? And they have evolved and adapted to the very different environments that we can find in this continent of Sahul, from deserts to tropical forests to mountainous areas and temperate areas. And they have managed as well to exploit the land in a sustainable way, that they could, live on this land and live very well for tens of thousands of years, basically, until Europeans arrived a couple of hundreds of years ago. So Aboriginal First Nations and Torres Strait Island First Nations and Papua New Guinea First Nations as well, they have developed some very good ways of surviving in the environments where they have evolved over generations, basically. And their genetic makeup is quite unique for that reason. And yes, they share a lot of their genetic information with the rest of the world, but there are genetic signatures that are specific to people who have, their ancestors living for generations here in Australia. Same is true for people in the Americas, people who stayed in Africa, and people who have evolved in the different regions of Eurasia. So basically, when a group of humans migrate and settle somewhere, usually it's going to be a small population, and then they're going to carry genetic information from the original larger group. And then when they settle and have babies over generations, they're going to build a very specific genetic makeup. And this is how we can distinguish populations around the world. Okay, so a European genetic makeup from the UK will be relatively different from a genetic makeup from someone living in Beijing, for example. When I say living, I'm not talking about people like me who have been traveling all around the world, okay? I'm talking about people who have deep roots in those particular geographical locations. And so the reality is that here in Australia, we have a range of very specific genetic makeups, because if you think about the time, so we estimate that, people have been around here for at least 65,000 years, if you believe the oldest archaeological evidence, and at least 50,000 years if you stick to the genomic estimates. But it's still thousands and thousands of generations of people living in the same place, okay? And developing social and cultural ways of doing things, and having like those identifying basically groups of people who are going to live at a particular place, and there's going to be marriage systems, and there's going to be eventually trading and migrations over some distances. But over time, what we observe is that in Australia, First Nations have a range of genetic diversity that is comparable to, for example, if you compare the top of Cape York and the Kimberley, you will have the same range of differences in terms of genetic information that you would observe between East Asia and Western Europe. So you can see that there's been enough time and enough generations to establish a very distinct map of genetic diversity across Australia. So in essence, people with indigenous ancestry, they carry some very unique genetic signatures that helped, their ancestors to adapt to the environment, basically.
Now, with mapping genes and migratory patterns in ancient humans, is there much we can learn about modern diseases?
Of course, when you change environment, when you move from place A to place B, there is a high chance that the pathogens, viruses, bacteria, parasites, might also change from that place A to place B. And when you land into that new place, maybe there's going to be a virus waiting for you, for which you have never developed any sort of immune defenses. And then it's going to wipe out your little group of migrants. Or a couple of you in that group will have randomly the right set of immune genes, and then they will be able to survive and then pass that resistance to the pathogen to the next generation, and so on and so on. So there is definitely a history of co-evolution between humans and their pathogens as people started to move around the continent of Africa or around the rest of the world. And there's a large range of pathogens out there, I can tell you. Most of them are going to be evolving, at the place where they are basically for very long times. Sometimes it's also going to be, due to the farming revolution, where some humans have started to live very close to animals, such as pigs and cows and sheep and chicken. And it's possible that pathogens jump from one species to the other. That is recorded. We have evidence for it. Or the other way around as well, from humans into those animals. And then you create what we call zoonotic epidemics. So there's development of pathogens through this constant contact between animals and humans. But that is so big with wildlife. Okay, I mean, bats, for example, a good example of being a reservoir for viruses that jump into humans, and other birds like wild ducks and stuff like that. Therefore, you have this constant battle between, the pathogens waiting for you in some places and the new groups of humans arriving in that particular place. Of course, there's particular habitats like tropical or subtropical environments that are very prone to be rich in potential pathogens. And inevitably, if you move from north to south or south to north, you're going to cross those regions as a group of migrants. So the story of humans is very well shaped by those crossings of tropical and subtropical environments.
Just as there are so many diverse pockets, just in Australia alone, there's many diverse areas we could go into. When we've got you, we might have to get you back again on the podcast, but I wonder, just in closing, Bastien, just shoot from the hip. Do you think we're seeing, when it comes to genetic trends, a move towards greater genetic diversity or away, sort of a more blending and more commonality between the people today? Just a few thoughts to finish off this episode.
Yeah, not an easy one to answer, to be honest. At face value, if you look at modern day human population, we're expanding. Okay, I mean, the population size is growing. And I don't know if you remember what you learned when you were in primary school, but these numbers have increased dramatically over time. So right now, I think we're in the 7 billions of humans living around the world, kind of. And that population still expands, okay? When the population expands, there's new variation that comes in that population through the process of mutation, basically. And usually, these mutations are going to be neutral. They won't have any particular role. And so they will just drift into the population until complete fixation or disappearance. But most of the time, they will just be like traveling through the population over time. And then there's those mutations that may have functional impacts, positive or negative. If it's a negative impact, it might be purged from the populations very quickly. If it's positive, it might be fixed in the population over time. Of course, I'm only talking about population size here. But if you really want these processes that I just explained to happen, you need to have random mating between people. Okay? You can't have cultural practices that restrict, marriages, for example, or you can't have geographic isolation between two groups of people. You really need to have a constant random mating of all people around the world, which is not happening. We know it. There's a lot of migration happening in recent times. Okay, I'm an example. You are an example as well. We are not First Nation people who have been living for thousands of generations in Australia. We're coming from other parts of the world, and we now live in this country that we call home. But, and it's true for many regions around the world, where you have this constant, migration fluxes, basically, of people coming from other places and settling and then meeting with the locals and then mating and having babies, etc., etc. But it's still quite restricted, if you want. So there's still pockets of populations around the world where there's not that much of this, what we call admixture or genetic admixture. And so the question of expanding genetic variation or reducing it is, I think it's a tug of war, kind of. In some settings, yes, I think there is an increase in genetic diversity in some settings, but in others, there's going to be a decrease of genetic diversity as well. Personally, I don't think the decrease in genetic diversity at the point where we are in the state of the genetic diversity of the human population is really good. And I think we're a relatively healthy population from a genetic point of view. But where my mind goes, usually it's more about the loss of culture, the loss of languages, the loss of, ways of being and thinking, the loss of many knowledges. That is not genetics, okay? It's entirely social, but that's happening. And there's some sort of, disappearance of a lot of richness in that has been established over the course of human evolution today. So I think genetics is probably not a big problem, a big issue right now. It would take a very, very dramatic event that would wipe out, 95% of the population to then start worrying about the loss of genetic diversity in humans. Yeah.
Wow. Okay. Well, I think the homework from that is you want us to mate across cultural lines, but protect as much cultural heritage at the same time as possible. We'll take that. Associate Professor Bastien Yamas, thank you so much for being part of This Medical Life.
Thank you very much for inviting me as well.
This Medical Life is recorded in the Talked About Marketing Studios in Adelaide. For show notes and more information about the podcast, visit thismedicallife.com.au. You can contact the hosts on social media. Dr. Travis Brown can be found on X. His username is @Dr. Travis Brown. That's Dr. Travis Brown. And Steve Davis can be found on LinkedIn. Go to linkedin.com/in/TheRealSteveDavis. This has been a PathNotes Proprietary Limited production.
Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award-winning Ozcast network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast.
Ozcast.
Simply head to ozcastnetwork.com for details.